ATAC-seq, or the assay for transposase-accessible chromatin with high-throughput sequencing, is a technique used to map chromatin accessibility across the genome. By identifying regions of open chromatin, it reveals potential regulatory elements such as promoters and enhancers that control gene expression and indicate regulatory activity. The ATAC-seq library is generated using the TN5 transposase, which simultaneously fragments DNA and inserts adapter sequences in a process known as tagmentation. TN5 preferentially targets accessible chromatin, resulting in selective tagging of open DNA regions. The adapters enable PCR amplification to generate sufficient DNA for sequencing while preserving the enrichment of accessible regions established during tagmentation. The amplified fragments are then sequenced and aligned to a reference genome. Regions with high read density correspond to open chromatin, whereas regions with low or no coverage are associated with tightly packed heterochromatin and reduced accessibility. Single-cell ATAC-seq extends this approach to individual cells, overcoming the limitation of bulk ATAC-seq, which averages signals across many cells and can mask cell type-specific differences. To carry out single-cell ATAC-seq, platforms such as the 10x genomics Chromium system may be utilized. In this approach, tagmentation is first performed on nuclei in bulk, after which nuclei are partitioned into individual gel bead-in emulsions, GEMs. Within each GEM, DNA fragments are labeled with unique barcodes specific to each nucleus. The barcoded fragments are then PCR amplified before sequencing, increasing yield while preserving cell identity. This enables chromatin accessibility to be profiled in thousands of single cells, revealing cellular heterogeneity within complex tissues. Although ATAC-seq identifies regions of open chromatin, it does not directly determine their functional outcomes. It is therefore often combined with RNA-seq to measure gene expression and ChIP-seq to identify protein binding or histone modifications. Together, these complementary approaches provide a more comprehensive understanding of gene regulation.